Hello guys, so topic for today is heme metabolism. We'll be studying the heme metabolism in several lectures. In heme metabolism, the various subcategories that we'll be studying is the heme synthesis, the heme degradation, heme synthesis, heme degradation, that is the heme catabolism, bilirubin metabolism or the heme catabolism. Then we'll be studying about the sickle cell anemia, the disorder related to this, and the various porphyrias, very important topic. And porphyrias, this is a very important clinical aspect topic. And various clinical cases that we'll be studying in this is the sickle cell anemia, already discussed, and then thalassemia, also related to hemoglobin in heme metabolism. Thalassemia is also related to this. Then porphyrias, different type of porphyrias that we'll be studying. To mention the names, these are acute intermittent porphyria, congenital erythropoietic porphyria, porphyria cutanea tarda, and then jaundice. Hemolytic jaundice is also related to this. Hepatic and hemolytic jaundice and obstructive jaundice. Three types of jaundice: the hemolytic jaundice, the hepatic jaundice, and the obstructive jaundice. These three types of jaundice. So starting with the heme synthesis, our very first topic, the heme synthesis. Heme synthesis. If you study the heme synthesis, basically the site, the most important thing, where it takes place. It takes place in the erythroid precursor cells and the liver. So it takes place in the erythroid precursor cells, and the second one is the liver. 80% of the heme synthesis takes place in the erythroid precursor cells and the remaining in the liver. And in these, some steps takes place in mitochondria and the other in the cytosol. For example, the very first step of ALA synthase that we'll be seeing abhi is the mitochondria में होगा, and all oxidases, जितने भी oxidases होंगे steps वो mitochondria में होंगे, and the remaining all the steps will take place in the cytosol, and the rest all steps will take place in the cytosol. So basically what are the steps in heme synthesis? There are basically 4 steps, 4 steps हैं heme synthesis में. सबसे पहले क्या होता है? Synthesis होता है porphobilinogen का, synthesis of porphobilinogen, PBG के नाम से मैं याद करेंगे, PBG. Then the second one, porphobilinogen की synthesis के बाद जो दूसरा आएगा, that is the conversion of porphobilinogen to uroporphyrinogen, conversion of PBG to uroporphyrinogen, UPG. And then third step is the conversion of uroporphyrinogen, UPG, to porphyrin. Yeah, protoporphyrin. We can say protoporphyrin. बीच में और स्टेप्स हैं. एंड देन थर्ड वन इज द इनकॉर्पोरेशन ऑफ Fe+2 इनकॉर्पोरेट होगा. तो दीज आर द बेसिक 4 स्टेप्स दैट कम्स अंडर द heme synthesis. Now first, in detail, if we see all the steps, how this takes place. So starting with the first step, that is, it starts with succinyl coenzyme A and glycine. So succinyl coenzyme A, you can write here succinyl coenzyme A plus glycine. Succinyl coenzyme A and the glycine, ये मिलके बनाते हैं तुम्हारा del amino levulinic acid. These form del amino levulinic acid. These del amino levulinic acid, it gets converted into it forms porphobilinogen. Porphobilinogen then gets converted to hydroxymethylbilane. Hydroxymethylbilane. bilane. Hydroxymethyl Now, this hydroxymethyl if we talk about the hydroxymethyl it cyclized to form uroporphyrinogen. Uroporphyrinogen, then it gets converted to coproporphyrinogen, then it gets converted to coproporphyrinogen. Coproporphyrinogen then converts to protoporphyrinogen. Protoporphyrinogen then converted to protoporphyrin, and then protoporphyrin में ही incorporation होता है Fe+2 with the help of ferrochelatase to form heme. So फिर से पहले steps पे अगर आ जाए हम, तो succinyl coenzyme A plus the glycine with the help of enzyme ALA synthase. ALA synthase requires PLP, and ALA synthase, it is of two type, ALA synthase 1 and ALA synthase 2. ALA synthase 1, it's present, it is inducible and it is present in the erythroid precursor cells, EPC. Now, succinyl coenzyme, इसमें से coenzyme A will be removed, the CO2 will be removed, and one group more, that is SH group, is also removed from this to form the del-amino-levulinic acid. Now, two molecules of del-amino-levulinic acid will form porphobilinogen, जिसे हम PBG कहेंगे. Now, this requires ALA dehydratase. It requires zinc as an enzyme, and it is inhibited by lead. PB is inhibiting this thing. Now, ALA dehydratase, zinc, dehydratase, that is something is removing water, minus 2 molecules of H2O is removed to form porphobilinogen. Now, porphobilinogen PBG, 4 molecules porphobilinogen, it is a monopyrrole. Porphobilinogen is a monopyrrole and its 4 molecule मिलके बनाता है linear tetrapyrrole. Hydroxymethyl is a linear tetrapyrrole. 4 molecule की जरूरत पड़ती है. ये क्या तुम्हारा लीनियर टेट्रापाइरोल है. इट इज अ लीनियर टेट्रापाइरोल, एंड इसमें से पोर्फोबिलिनोजन से बेसिकली 4 मॉलिक्यूल्स अमोनिया बाहर निकलते हैं. सो इसमें एंजाइम क्या होगा, पीबीजी, दैट इज पोर्फोबिलिनोजन डीआमीनेज कह सकते हैं, और वी कैन आल्सो से एचएमबी, हाइड्रोक्सीमेथाइल बाइलेन सिंथेज, or one more enzyme is the uroporphyrinogen 1 synthase. Because this is also known as, this thing is also known as uroporphyrinogen 1. Now this hydroxymethyl bilane is converting into uroporphyrinogen 3 with the help of uroporphyrinogen co-synthase. With the help of uroporphyrinogen co-synthase, it's get converted into uroporphyrinogen 3. अब इसमें क्या होता है, इसमें से यूरोपोरफाइरिनोजन 3 को सिंथेज, बेसिकली नथिंग हैपन, यहाँ पे केवल साइक्लाइजेशन हो जाता है, लीनियर गेट्स कन्वर्टेड इनटू अ साइकिल. नाउ यूरोपोरफाइरिनोजन का अगर हम साइकिल देखें तो हर एक तुम्हारा पाइरोल है. तो दिस इज ए पी एंड पी ए. दिस इज द स्ट्रक्चर ऑफ यूरोपोरफाइरिनोजन. Okay, this is the structure of uroporphyrinogen. Now, uroporphyrinogen 3 is इसमें uroporphyrinogen decarboxylase लगता है. uroporphyrinogen decarboxylase. इसमें से क्या होता है, तुम्हारा CO2 molecules will be removed. 4 molecules CO2 का निकलता है, तो तुम्हारा coproporphyrinogen 3 बन जाता है. And if we say the structure of coproporphyrinogen, this is MP, and then we have PM. This is the structure of coproporphyrinogen. Now, coproporphyrinogen पे लगता है तुम्हारा coproporphyrinogen oxidase. Coproporphyrinogen oxidase make it convert into protoporphyrinogen. यहाँ से तुम्हारा 4 molecules CO2 is again removed. And then protoporphyrinogen is formed, which is nothing but this thing. Protoporphyrinogen, we have not much change. Vinyl group is adding in here two places, MV, methyl vinyl, and then we have methyl and propyl again, propyl and methyl again. So this forms the protoporphyrinogen. Next we have the protoporphyrinogen oxidase. Protoporphyrinogen oxidase convert into protoporphyrin, and then we have the incorporation of Fe+2 with the help of enzyme ferrochelatase to form the heme, that is our final product. So this is all about the heme synthesis. इसमें the first step, the very first step is only the rate limiting step. That is succinyl coenzyme A and glycine get converting into delamino-levulinic acid. यही feedback inhibition देता है. This transport of delamino-levulinic acid, यही तुम्हारा first step is only the rate limiting step. Revising very quickly, we have succinyl coenzyme A. Succinyl coenzyme A, glycine gets converted into delamino-levulinic acid. Delamino-levulinic acid gets converted into porphobilinogen, which is a monomer. It gets into polymerized to form hydroxymethylbilane. Hydroxymethylbilane cyclizes to form uroporphyrinogen 3. Uroporphyrinogen 3 gets converted into coproporphyrinogen 3, then protoporphyrinogen, and then we have the protoporphyrin, and then at last we have finally the heme. And the enzymes which are related to this is, we have first of all ALA synthase, then conversion this takes place with ALA dehydratase. Porphobilinogen to HMB is by the PBG deaminase because ammonia is removed, or HMB synthase because HMB is formed, or the uroporphyrinogen 1 synthase because this is also known as uroporphyrinogen 1. Hydroxymethylbilane, then uroporphyrinogen 3 co-synthase, and then we have 4 molecules CO2 removed, so uroporphyrinogen decarboxylase. Synthesis of protoporphyrinogen may coproporphyrinogen oxidase, then protoporphyrin is formed with the help of protoporphyrinogen oxidase, and then Fe+2 is incorporated with the help of ferrochelatase. So this is all about the heme synthesis. Takes place in the erythroid precursor cells, EPS, EPC 80%, and then mitochondria may ALA synthase and oxidase step take place. So we have covered the heme synthesis topic. In the next lecture, we will be discussing the heme degradation or the heme catabolism. Thank you very much.